So hopefully I'm audible and visible. So this is week 4 live session for the introduction to proteogenomics. So in this, in previous week we had discussed the different how the in the proteomics mass spectrometers have helped in the evolution of the field of proteomics and have led to the identification of the different help in the identification of different proteins in a high throughput manner, and how the different mass spectrometers, what are their roles, and how uh, one can identify and analyze the different target proteins, and identify the different proteins in different samples. So now in these sessions we will look a little more into that the generated, and how those can be analyzed to understand, which are done by your different software, that how they analyze the spectra and how those masses are calculated and how the peptides are finally sequenced, and which are used for identification of the protein. So as you see that this is the chain of a peptide with a C terminus and the N terminus. So whenever, so these are the, so the fragmentation patterns, that how the peptide is fragmented depends upon three factors. So first factor is the primary amino acid sequence. So since we know that the all the amino acids have different side chains, and that what it makes the different, gives the different properties and the charges and the to the protein and to the amino acid, that makes it unique. So that depends. So the primary amino acid sequence will have an effect on the fragmentation pattern, followed by the internal energy and the charge state. So this is how, if this paired, so if the the peptides can be detected, can be possible only if it carries at least one charge. Without a single charge it won't be able to get detected. So if you see that the charge retained on n terminals, so whenever the charges, so like nearest to the N terminals, it is known as in the form of ABC ions, whereas the charge retained on the C terminals are known as the XYZ ions. So that's why the nomenclature came. So this fragmentation pattern, so the Y and B will come as a pair. Similarly, X and A, and similarly Z and C should come as a pair whenever a fragmentation happens. But so this is how the structure of a singly charged sequence would look like. So this is how the A, so the ions, what we told, that A ion, B ion, C ion, XYZ ions. So how they look like when a single charge ion. So how a singly charged ion will look like when it is fragmented. For example, how A ion will look. So how an AIN looks, the AIN looks like this when it is fragmented with the charged on at the N terminal. Similarly, is the, this is the B ion. So with the side chain when it uh, so these are like the R1, R2 are your side chains, and when it gets fragmented, so it produces a signal at the in terminal. Similarly, whenever this is the C ion fragmentation after fragmented, and this is the X ion, this is the Y ion, and this is the Z ion. So hopefully by now you all are familiar that what we are looking at for the different charges are different ions that are generally that are being released or identified in a mass spectrometer is our Y ions and the B ions. These two ions are, and among which Y ions are most prevalent due to the our use of our proteolytic enzymes, and the way it is fragmented, it provides, it favors the fragmentation in the Y ions, which are detected to finally identify the amino acid sequence. So these are all the singly charged ion species, how it looks like in the fragmentation when it gets fragmented from a particular peptide. But in electrospray ionization, those fragment ions are generated usually carries one or more charges, and that's why electrospray ionizations are more favored with giving a narrow M by Z range or a small M by Z range to be detected by the in the mass analyzers. So this is so these structures are somewhat uh, i- important to understand in a way to visualize that how your peptides are getting after you make the peptides, how that is getting separated and then getting fragmented, and then you are trying to identify those particular so these from these ions are getting detected, and how we were trying to find out the masses, and then finally detecting the amino acid sequence and then making the protein sequence out of it. So these are your singly charged ion species, how it is looks like. So among which, so this happens when your fragmentation is proper and you are getting a proper A ion, B ion, C ion, different ions. But there also sometimes happens is that it's a concept of the internal cleavage ions, because all your amino acids and all side chains are not at all favorable for the proper fragmentation, and also it depends on many various factors. So there is the internal cleavage ions. When it is formed by a combination of B type and Y type cleavage, it produces amino acylium ion. So this is looks like something like this. So this is a combination of your B type and the Y type ions. So this this is known as your internal cleavage ion. Similarly, is another type of ion concept is the ammonium ions, which is that a single side chain formed by combination of A type and uh, Y type. So one is your internal cleavage ion, which is formed by a combination of B type and Y type. So and another is your A type and Y type. So uh, so it what happens is that in the previous previous week, actually we had discussed regarding the spectra, that the CID and ETD. So generally we are using CID, collision induced dissociation, so it can produce some, so this uh, what masses it should look like. So ammonium ions, there is a specified this is how ammonium ions would look like, and for each amino acid, so it looks somewhat different when it is actually to be fragmented. So then the you need to, or the software needs to identify particular those values which shows the ammonium ions for a particular amino acid. If it gets detected for that particular mass, uh, if you get that, then you will understand that it is the ammonium ion, and you need to assign that amino acid accordingly. So this is it provides some compositional constraints on your sequence assignment. So another ions are your satellite ions. So satellite ions are those when the CID, collision induced dissociation, is formed at high kilovolt energy. So not only your XYZ ABs or the ammonium ions or those ions are formed, there are some additional ion types. So on the basis of your side chain, those also can be generated uh, on your in your system. So these are known as your satellite ions. So what are those? So these are represented by D ions, V ions, and W ions. So now these D, V, W are some extra ions on that are generated out. So these D and W ions are also quite helpful on differentiating your isobaric leucine and isoleucine. So why this? So how it helps? So in your leucine and isoleucine, what happens is that it is mostly the change in the side chain. Otherwise your everything is same. So due to the generation of this kind of ions on the basis of your side chains, so you can, what you can do is that you can help differentiate your leucine and isoleucine as well. So this is one kind of help it can generate, but these are mostly additional ions that are generated. So this is satellite ions. So this is how you will generally it is calculated the fragment ions of the M by Z values. So A ion type A is your uh, this one neutral M. So N determines your N terminal, C determines your C terminal amino acid mass. So A minus uh, NH3, A can be also H2O can be a loss of H2O can also be there. So if you found out one mass of an monoisotopic mass of something minus 18 for H2O, then you have to understand that that particular amino acid have lost in the water H2O have been lost, and then you can find out. Similarly, for the B ions, and then partial side chain, and then XYs and the Z ions. So this is so whatever in the ABC we'll find out in the N terminal, and whatever for the XYZ we'll find in the C terminal. So this is are the some of those ion types and how to calculate their M by Z values, which will help you. So now if in your mass spectra two peaks appear which are having 28 Dalton apart, so you need to understand that these are constituents of your A and B ions of the same peptides or the same peptide species. So when you will find in the masses coming as 28 Dalton apart, so these are your A and B ions. So A and B ions always come up 28 Dalton apart. Similarly, if the two peaks are equidistant from each other, then you will understand that it is your Y and B ion pair. So from there you will calculate accordingly Y7 minus Y6 and here B1 minus B2 for example. So you will get that what are those. So this is are the some general ion types and how your uh, what are the masses that are calculated. So this is how as I think you all have seen in the previous mass course hands-on as well, that these are that how the peaks are generally identified. But generally what happens is that in the software it already gives us that what are the different peptides and the different these different peaks correspond to which ions, and it calculates the masses and derives the amino acid sequence, and then it maps with the protein sequence and helps us in the identification of proteins. Otherwise, if it was to be calculated on the basis of your in a manually, also you can interpret the spectra. Once you got it, then you need to have looking at the pairs that okay which one are equidistant, then why that's our Y ions and that's the B ions. Then if these two are having 28 Dalton, then it's your A ion and B ions. Then if you there is a loss, the mass monoisotopic mass that you are getting minus ammonia, then that is for that amino acid less ammonia, then the amino acid less than water. So that all things you need to calculate manually, and then you can identify those masses. But with the help of software we can easily do that, and it helps us in the identification of that particular protein. So this is the reason why loss of water from B ions is due to the presence of your glutamic acid at N terminus. So if glutamic acid is present at your N terminus, then generally what happens is that, so these are some of the characteristics of your peptide or of the basis of the amino acid. What happens is that there are some changes or the sum of those losses that happen. And so this is one example is that your loss of the water from B ions due to the presence of your uh, glutamic acid at N terminus. Similarly, losses of ammonia happens as a result of glutamine in the N terminus of the sequence. If glutamine is present at the N terminus of the sequence and the fragmentation happens, then losses of ammonia generally seen, which gives you the particular result. So this is how mostly the fragmentation happens, and fragmentation are spectra are generated which are taken forward. So why this is also necessary to understand that, which we will look into the further during the data analysis. So all these correspond to certain factors that we need to keep in mind when we are doing the secondary data analysis or trying to identify particular peptides or proteins that are upregulated. So now if we need to look at to try to understand that how a mass chromatogram looks on. So if we try to understand that what are the information that can be interpreted from a mass chromatogram, are that it gives the total gradient. So for example, here is that it is from 0 to 120 minutes, means from 0 to 2 for the 2 hours it was the peptides were eluted out based on the different gradients, and then it was detected in the mass spectrometers. So why we need to elute out the peptides, so that the peptides can come out and the number of ions are generated. All the ions generated are not bombarded at the mass analyzers and the mass detectors at once, and it can be easily it can get sufficient time to detect all the peptides and all the proteins that are present in our sample. And also it depends on other factors like your flow rate and all those which will help you to get the proteins get the identified the maximum number of peptides and the finally the maximum number of proteins from your sample. But here the concept is that the a good chromatogram should be a Gaussian in pattern. So why Gaussian in pattern? So we will when we will look into more deep in the parameters and all, then we can understand that why it is required. So the peptides mostly that when we will get a good coverage is that the peptides having the majority of the intensities in the middle, and the illusion start point and end point should be very much smooth without having any humps or the distorted peaks. Then only we can get a very good amount of peptides and the MSMS spectra being detected by the mass spectrometer, which will give us a good result. So now what happened with this chromatogram? So here you can see that the illusion started, though it started a bit early, and also it happened is that the illusion did not continued as it was, and it ended abruptly here without giving any such pattern of the Gaussian pattern or any such thing. So from here only you can understand, if you compare this chromatogram along with these two chromatogram, then only you can understand that the number of proteins identified or the coverage would be much less. So from here, so if you are running, for example, if you are running three control samples and the three treated samples, and in between it happened like this, so it can be issue with the column, issue with the sample. So anything can be there which cannot be interpreted uh, just by seeing the chromatogram, but surely there is some issue existing. But the problem is that, so all these factors lead to you lead to difficulty in the data analysis when you will do data analysis and try to incorporate those. So all these factors will add up for some of the difficulties that you need to address while performing the data analysis. So this is how this chromatogram isn't showing a good. Similarly, this chromatogram you can see, so that it is not showing any dense chromatogram or along with any Gaussian pattern. So it is being eluted on and off, and it is uh, going on. So sometimes, so this is how you can uh, try to understand the. Sometimes it also happens that your one single peak suppresses all your chromatograms, but generally the illusion pattern should be uh, quite good. So not like this. So firstly, that reason why we need to, why we are stressing out on that it will be on the Gaussian pattern or how the peptides, because the peptides, the way we elute. So that generally, if we take that the if you are using a coupling your mass spectrometer with a nano LC or a nano liquid chromatography. So your flow rate will be in your nanoliter per minute. And then what you are doing is that you are trying to eluting, as we mentioned, that we use a reverse, as we had, I think, discussed in the first way, that we use a reverse phase liquid chromatography. So in the reverse phase, we have the stationary phase. We are binding the C18 columns with our hydrophobic peptides, and then what we are trying to do is that we are eluting out the peptides using more in the mobile phase, having uh, uh, hydrophobic solvent, and then we are increasing the gradient as it gets eluted out. So here you can see that what we are doing is that we are gradually increasing our gradient of the hydrophobicity, so that the peptides on the basis of their hydrophobicity gets eluted out as the from the stationary phase as the mobile phase flows out. So here, from as we increase the hydrophobicity, and the peptides gets eluted out. So that's why the majority of the peptides that are will come out should be, we should expect within the near the mean region, or near the having the maximum outflow. And during the start of the elutions, the hydrophilic peptides that should not actually you phase when we are just uh, upgrading the gradient, just increasing the gradient, that should not elute out. So that's why the starting point of the illusion and then your ending point, it should be smooth and smooth curve. So this is an ideal gradient that should give you a good chromatogram with a full Gaussian pattern, and then you will get the full maximum coverage out of your protein. So the elution of peptides are occurring in the range of the less hydrophobic, then it more hydrophobic. As the gradient is increased, more hydrophobic peptides are eluted out. So this is how the, so that's why we are stressing on the fact that it should be more of a Gaussian pattern with and the peptide should be detected in that range and those intensities. Yeah. So any question, anything till now? No, right. Yeah. So if you no questions, I'll just, yeah. So I'll move up. So moving on. So this is another concept of what is known as your dual volume. So that is that system volume from the point at which your mobile phase solvents are mixed. So until they reach to the head of the column. So as I mentioned you regarding that, the so your solvents are generally mixed and your hydrophobicity gradient is uh, made out. So now if you look at the, sorry, just, yeah, so if you just look at that, how your arrangements are made. So here your solvent that you have kept here, which is your mostly your water and your acetonitrile, will be mixed and in the pumps, and then it will reach to the column. So the system volume from the point at which your mobile waste solvents are mixed until the reach of the head of the column, it is known as your dual volume. So it comprises your mixer, then connecting tubing and auto sampler loop. So this is how in a high pressure mixing LC, what the how it is mixed. So what is the reason of putting it here is that it also plays a role in your whatever gradient and whatever your chromatogram you saw. So this also plays a role, and it also gives you the, it can also give you slight impact on the eluting certain peptide with certain characteristics. So like is that so with the connected with the dual volume is the dwell time which is obtained by your dual volume by flow rate. So if you have a dual volume of 3 ml run at a flow rate of 1.5 ml per minute, so you will have a dual time of 2 minutes. So delay created by the dual volume can make your difference in the for the different gradient systems that you will make. So that will help, that will have an effect on your created by your dual volume and the dual time, but it will not have any effect on your isocratic flow. That is when your solvent composition is same. So if you are using the same solvent composition throughout, then you will not get a good gradient, or you will not get a not no gradient is there, so you will not get a good chromatogram, but it will also do not have any effect. But whenever you are using an increasing gradient or the change in the gradient, so then definitely you will this will also have an impact on your system and how the peptides are eluted out. So this is another concept which helps you in understanding that there are some issues or the errors with the uh, chromatogram or the change in the gradients with or identifying certain characteristics of the peptides and those. So all these factors you will be better understand and better appreciate when you will go and see the data analysis techniques, that how all these steps can incorporate all these difficulties and can the starting from your fragmentation pattern to the good from getting the chromatogram, then the uh, components of the system, columns getting old as you run the samples and all. So all this creates your gives certain artifacts or the technical variances in your samples whenever you run. Along with the there are some uh, the along with the noises are also not noises, but generally the way the algorithms work and the peptides the proteins are identified also plays a role. So for example, now if you look at it in these different chromatograms, so now can you identify that which chromatograms are contaminated or not? So you should do it. So now we have said that the illusion and the end of the illusion is quite important for us to understand that how the particular run has happened or the particular chromatogram looks like. But now what we can see here is that after the so since you have seen the gradient as well and could understand that the more hydrophobic peptide should elute out, and then there should not be any hydrophobic peaks or something that should come out. But here you can see that there is a hump or your distortedness in the while there is an end in the illusion. So something means that the something is coming out from your column and diluting out, which is not expected at that contaminants that you need to that will indicate that you will not get good runs if you with this chromatograms. You need to clean up your system and then you can uh, run and line up. So this is how you can understand your different contaminated peaks by if you look in the end of the chromatogram if there is some any issues or any problems, and then can work or work on that accordingly. I was telling
So
that
how
till
can we
now I hope
identify
you all are
this is
clear with that
contaminated peaks?
proteomics experiment. So
Hello?
you are taking a sample, might be it can be clinical sample. You are collecting your sample,
Hello.
then you are processing your sample, then you are doing your after
Hello,
digestion you are
Ankit.
doing the desalting, and after desalting you are doing your in the separating out in the LC, and then you are taking out the instrument. After which what we saw is that today that there are fragmentation and all those mass spectrometers and all ions are calculated. Now what we could see is that we are getting different types of chromatograms or the different types of events. So if the same sample of the different, if you are running and one after one sample your column is getting choked or your tubings are getting or your instrument performance is getting down, so what is happening is that you are getting a different patterns of your chromatograms. But if you're now if you need to have your
Hello, Ankit.
sorry.
Hello, Ankit.
Okay, just second. Hello,
Yeah, Ankit, can you hear us?
I'm
Hello.
audible, right?
Yeah, you are audible. Can you hear us? Hello.
Okay.
Hello,
Is
Ankit.
there any question or something? One second.
Hello.
Hello.
Can you hear us?
Yeah, I can hear you now.
Yeah. I think there was some issue. Somebody was
Uh,
asking questions to you.
yeah.
Whoever is having questions can ask now. I think Ankit was facing some technical issues.
Sir, can we please go last slide?
Last slide. Okay. Yeah.
Sir, I am asking how can we identify contaminated peaks? Hello?
Yeah. So you are saying that how you are identifying this contaminated fix, right?
Yes, sir.
Yeah. So this contaminated fix, as I just mentioned, that if you in this part of this region of this chromatogram, so here you should not get any kind of illusion out or something. So here it means that something other material which is much more hydrophobic than your peptides or something or some carryover thing which is suddenly eluting out from your column and those things. So that's why you will say that it is your contaminated ones.
Okay, sir, it's
My,
all right.
yes, I don't know if there was some issue. So yeah, yeah. So just let me know if I am audible.
Yes, sir, you are audible.
Okay. Yeah. So as so I think someone has written in chat to again repeat about that. So what I was telling is that as we have seen that uh, in our gradient that how it should be eluted out. So in this zone there should not be any dilution, as most of our peptides should have eluted out before only. And so if there are certain peptides or certain things which are more hydrophobic than your peptides, or if certain peptides have been there in the carryover and it gets eluted out suddenly, then you will get those distorted and this those type of things which you need to which will showing that it is a contaminated and it goes off after cleaning up your systems. I'm clear, right?
Yes, sir.
Okay. So moving on, so if there is uh, no more questions.
Ankit, your voice is coming a bit slow, like a bit low.
Okay. Yeah, now.
Yeah, now it's better.
Okay, okay. Yeah, so I'm doing like that. Yeah, sorry, so there were some issues. Yeah. So now, as you could see that there are so many places where your data can have noise, your data can have artifacts, your data can behave differently. So now what we is that we are trying to compare your two samples, two sets of groups, that your control and your disease, or your control and treated or anything. So now what we are getting is that we need to minimize the noise that we are generating, or in a way it is getting generated, and without affecting our real signal. So firstly we have to understand what is the noise and uh, what is your noise and uh, what is your signal. So if there is your so now so the data normalization is our technique by which we are trying to, or we will try to minimize our noise from the data, and that doesn't affect the signal. So this is done to tackle or minimize our variations in the experimental conditions and the data acquisition. So whatever, so as I told, that these are different steps and the different processes are there. So we need to understand that all these variations that can be introduced in each step. So we need to minimize those. So this is how we can adjust the scale of values for. So we need to also adjust the scale because we are trying to compare. And then the major concept or main thing is to minimize our technical variation and extract out the biological variation. So hopefully that it is clear with everyone, and the pictures of the chromatogram and all have made it visually clear to every one of you that how that in the real data sets, when you run your samples, there are really you will get some kind of data which are coming or behaving differently, and hence we need to try to transform those data, and then only we can perform the analysis. So hence that's why, because before going to the analysis steps and the techniques, why it is important to understand that why it is required. So I am hopefully that you all are clear now that why we need to go for pre-processing before we can do the statistical analysis to understand the differences. And also we need to understand that how our data quality is, and how our run is, and how good the proteome coverage is, and then only we can try to find out the uh, what are the differences. So this is, so in the data normalization thing. So now, as I have mentioned, that you all have also seen that the data distribution from the chromatogram. But then after the chromatogram, so here if I just give a brief description. So once you have done your data pre-processing, means data has been run and you have got the raw data, then you have done in the as a kind of a primary data analysis. You have tried to identify your peaks and identify your peptides and your particular proteins. So now on the basis of your, if whenever a data distribution, if you take out from a population, a sample from a population, so it tends to have a kind of your normal and Gaussian distribution. So this is not the one that you have in chromatogram, but when you have got the Excel sheet with the peptides and the proteins, with the identified peptides and your proteins, so that values and the intensities of that also on the basis of your sample would look like to behave behave and distribute in a normal way or in a Gaussian distribution. So now you can see that you have run three different samples or your three different proteins. So that three distributions are like this. So if you get three distributions having a different standard deviations across the mean and having three different means across the distribution, so then you cannot apply the statistical tests to identify or compare the means between the two groups. So now what we can do is that trying of a normalization is that the centering. So what is that centering? So you try to subtract the variable mean or median from each values of that same variable, so that it gets to the shifted towards the median, or it becomes centered to the median. So this is, in a way, it is also known as the linear rescaling. So since you are just subtracting from the average and median, so it will come across the group. So this is how uh, the data, then it will become ready to for you to compare between the two groups. So this is in a real and ideal data set. So not an ideal, but in a real data set, what happens is that this distribution, so this is an example from a proteomics data set only, so that how you get a distribution which you assume, and after the scaling, that it follows a kind of a Gaussian distribution that will lead you to apply your statistical test. So that's why, so another important thing that needs to be we need to keep in our mind is that in making the things shifting or centering or making a proper Gaussian distribution curve. So since we are here aiming for to identify our differentially regulated proteins, or to identify the proteins or the markers here, so that stays in your end of the tails, or because in the distribution, the majority of the distribution that follows within this are unchanged. That is your most of the proteins that will be in your samples will be is same concept, same lies with your gene expression data as well. So if your gene expression or protein expression, most of these things are unchanged in your both groups, but there are certain proteins or groups that are staying outside, and that is making your distribution looking different for each sample in a biological way. Apart from the technical way, the biological variations are also leading you to your distribution to be behaving in a different way. So we need to be careful while applying any kind of normalization technique, so that our signal or the biomarkers that are present at the near the end of the tail, which shows the maximum your fold changes, or that will have your minimum p value, that should not be gets affected while the process of normalization that we are doing. So if we, after we have done the normalization, so generally what we do just in the proteomics data set to keep the originality. So in this manner of just reducing the noises, we cannot have the loss of signal as well. So in this trade-off, what we do is that we generally use for or go for median normalization to to use to does not affect the signals and helps us in get the required reduce our technical variation as usual. So now after this is the scaling. So what is the scaling is that, so as you can see that it is following a different standard deviation. This distribution has a different standard deviation. It is more widespread, one is narrower spread. So these all these things can happen in our real data sets when we have the different samples run. So if you have, after you run your one sample, then your column got choked, and then again you injected your sample, though it did not show any such variation that you cannot take those two data sets, or you have taken all the necessary precautions, but the distribution looks quite off, and with the shift in your little bit the mean and the variance, there's a shift in the variance. So what you can do is that you, after normalizing and bringing the medians equal, then the scaling can be done so that the distribution follows the same standard deviation in all the things. So that's called your scaling. So generally what you can do is that you can divide your values by standard deviation or median absolute deviation. So if you do this, then your distribution will come all your values, that all your samples will be following the same within this, it will, the range will fall within the same standard deviation. So this is how you can do. So median absolute deviation is that one which determine your spread of data, and when your data is not normally distributed. So once you have done your centering, and then if you perform the scaling, so that procedure is called your standardization. So whenever you see that some data sets or the data has been standardized, so what you will say is that the standardization means that it is been centered as well as it is scaled. So our data is centered means we are medians centering has been done with all the means lying together, and then it has been scaled. So it will give you a median scaling. So another so how many, what are the different types of scaling are there? So z scaling. So if you do your data set as z scaling, so what will be is that it is median centering with the and divided by your standard deviation to perform a standard deviation scaling. So that is your z scaling. And another thing is median centering with the mat scaling. So mat is your divided by the mean absolute deviation. So this is how you can perform your, you just reduce your technical variations or biasness. So now these two are these two are generally used in our proteomic data sets, but now there is one another technique of the normalization that is mostly used in your microarray data sets or your RNA-seq data sets. If you do it, so this is a normalization. It's known as your quantile normalization. So what we are stating here is that we want to make sure that our technical variations or biasness or whatever changes during the whole clinical sample or any sample in that manner, uh, collection, your processing, then your data acquisition and everything. So now I want to reduce my technical variation, but I do not want to change the loss of the signal. But here in this quantum normalization, what is does it does that it tries to, it makes the two distributions identical in its statistical properties. So what so this type of normalization cannot be is generally not used in our proteomics datasets, because in our proteomics dataset there is no single range within which a protein intensity should be measured, or we do not know that our protein intensity should come within this range only, like within 0 to 1 or 1 to 100. Then okay, so then what is happening is that we have our reference data sets or reference thing. Then what we can do is that we can match it, and then we can try to make it as that by taking the dividing the division between the two values, and we can increase that range. That okay, the performance of the instrument was down, and then we are just adjusting it to it. But since it has does not contain any fixed number, if we try to bring one reference and take one group as reference and try to do that, then we can lose our biomarkers. Why is it so? So I also have mentioned one line that most of our protein or remain unchanged, or only those at the tail of the distribution are the ones that would be like to have more biomarker effect, or would be going to have the large highest fold change or have the lowest p values. But when we are doing a quantile normalization, so it will look at those particular proteins which are having the maximum changes in this way. So it will just rank it that, for example, here you have the data set 1 showing 5 and the data set 250. So which one is, it will rank accordingly, and then it will give a normalized value. So for example, if your proteins having features which one is highly upregulated and less, so it will also get in a way adjusted to it, so that it becomes looks like identical in your statistical properties. So in this way it will give a huge loss to your data, and uh, it will have a huge effect on your biomarker identification. So that's why quantile normalization is not used, but in your RNA-seq data or the microarray data, quantile normalization works well and is used, because they are a fixed reference of number within which a gene expression is there, it is measured. So that's why quantile normalization is used in that. So it takes your true distribution as a test and a reference, and it tries to make it identical in its statistical properties. So this is so that's I mentioned that we need to protect our signals at the end of the distribution. So if the those remains mostly unchanged, and so in this end, and also if you know a little bit that most of the data distribution comes, if for a normal distribution, so within 3 standard deviations. So that's why we have measured your 3 standard deviations. So in the within 3 standard deviations only, all your data points lie in your normal distribution. So that's the case. So that's why we need to protect our signals. So those which are different will be generally following uh, that, and especially from after 1.96 standard deviation, mostly our values remain, which will give you that in the confidence interval of 95%. After 95% confidence interval, what we will get are those proteins which are having the differentially regulated. So that's why we need to protect our signals at the end of the distributions that are having differential expression in the two groups. And for this reason, we cannot use any for making our data much more robust or much more minimize the noise. We cannot have lose our data in this manner. So now another thing is the another having the two component Gaussian two component normalization, or the two component Gaussian mixture models. So this is your probabilistic model for representing normally distributed subpopulations within an overall population. So what here it is changed, that as I told, that it makes use of the concept that the some proteins do not change, and the only few proteins are there which changes and which produces the result. So it doesn't require to know which subpopulation of the data set belongs to. So it will, what we will do is that it will do the clustering. So you have the both control versus control and the disease, but it will group it accordingly, that proteins that are changing and proteins that are not changing. So then what you will do is that you will ask that, okay, so since there are, you have identified the proteins that are not changing, so you will perform the normalization only the with the proteins that are not changing. But here there is a so it looks like a lucrative type of normalization, which shows that, okay, so we are normalizing only with the proteins that are not changing, and our proteins that are changing are as I mentioned, that we need to protect the proteins that are having the differential expression in the different groups. So it is getting well protected by those proteins are well protected by that. So the proteins that are not changing are only getting uh, uh, will be doing the normalization, and all those proteins or signals will be there. But there are some problem with this kind of normalization, is that there are not a single very factor, that the only biological factor is not responsible for experiment. So as I was mentioning that, and in the fragmentation pattern, so why it is important to understand those your fragmentation pattern and the ions that get generated. So since we were saying that some amino acids residues leads you to your uh, cleavage ions, or there are some fragmentation issues, so that particular peptides or the during the experimentation part also might not get detected, or the peptides might not get detected, or your proteins not get detected. Then what will happen is that you will not get uh, that changes are not due to biological changes, but that changes are due to the technical changes. So then the two component normalization you cannot use. So that's why the generally what is happening is that the median mat normalization, and also it doesn't does a lot of changes, as well as it doesn't affect your, it doesn't also remains on your sample size and all those things. It doesn't remain dependent on it, and it helps you to reduce your technical variations and the biasness. So generally what we follow for any proteomics data set is median normalization followed by a mat scaling, which is known as your median mat scaling or median mat normalization, that is generally being followed. Yeah, so that's why. Yeah, so uh, any question till that for the data normalization, why we do it, and how to decide on. So actually the normalization depends on various factors like your data distribution and those mean variance and all. But in the proteomics datasets, we need to look at consider other things as well before selecting on for any normalization kind of technique. So any question till here?
Sir, could you just repeat the points that you said about median mad normalization? Why they are used?
So median mat is generally used because it follows, it doesn't affect our signal too much. Rather, it just does a centering with the data that are in the middle part and not changing, and then it does a mat scaling. So it's just it will make your within the range of the scale of the standard deviation, so that we can try to map between the means. So it is not affecting as quantile normalization is doing, like it is making your test data set as also your reference data set, which will lead you to a loss of the signal or a kind of a destructive normalization. Or also the changes, as I told, in the two component normalization, like there is a that the all the changes are not only biological but technical variances are also there. So that's why it is mostly used by the median mat normalization with no such stringency on your data, but also it will help you to correct the technical variations and the biasness.
Thank you, sir.
Yeah. So one second. So no question, right? So fine. So I will just. So hopefully you all are able to appreciate till now the reason to understand the fragmentations, then the how the illusion pattern, and then how we are taking out the data, and then we are trying to uh, do it the scale and normalize our data, so that we need to find out our some changes. So now moving on. So now data normalization can help us in. So for example, you are running, you have 50 samples and you are running an LFQ experiment. So hopefully you all are clear that what is the difference between LFQ and those. So LFQ is nothing but just we need to run each sample one by one, and on the basis of our uh, illusion pattern or by the spectral intensity count, we count our data, or we quantify our data, absolute quantify our data. But now if certain things happens is that your data gets you are running your data after 3 days, you have continuously run your data or your so first so so you have run your data, like the disease samples, and then you are uh, so you have I am hoping that you all have randomized your samples. So you haven't done all your disease samples at one and then running the control samples. So you have randomized your samples. But still after 3 days, what happens is that so you have run your 5 disease samples and then run your 5 control samples in a set in a randomized manner. So then what you are doing is that after 3 days your column got choked or something issue happened, or you could not process your samples, or something happened, and then after 3 days again you run. So then what you are getting is that the same disease or the same control samples. So if you look here, so you will getting a different value. So these are not your control versus disease or something that are different, but your samples sets only that what you have run, your control disease and your control disease are showing 2 different batches. So now you can clearly understand that what any difference that you would find out. So if you try to find out any difference between here in your two groups or the two things, here you will not get any relevant biological changes or the biological things. If you get also the changes, you won't be able to say that this is all due to my biological effect. So all this can be due to the technical effects, because you have run your samples and after 3 days there you can see a batch effect has been you can see a very good amount of visualize your batch effect. So what is that? The next first step is to do a correction of the batch effect. But the one thing that we should keep in mind, or we should understand, that the batch effect correction leads us to have a quite generate a quite big number of artifacts that can give us a false signal. So this all leads to the generation of the false positives in our data sets. So firstly, the first and foremost, whenever you design an experiment like this with having so much multidimensional, so in that which requires various steps, and you want to acquire the data and all this data of high scale, the first thing that should be done is your randomization of samples. So all your samples that you run should be in a random manner, and it should not be that one set of groups of samples are run first and then another set of samples. Then it will be detrimental and your experiment, and you cannot gain out your successful you cannot yield out successful results. Because if you have run one set of samples and then something happens to the instrument, and then one do the another set of samples, then whatever you will be testing out, it will be completely different, and you can never try to identify out or point out the differences, and cannot prove the differences to be biological. So what is the difference between the normalization and the batch correction? So I think this question should be raised in your mind. Is that one is that the both does the corrects the systemic technical biases, but normalization is quite of a kind of does within the samples, between the samples, but here it does between the group subsets. So here in the normalization it does through the sample wise, and it does just the which does not give a quite a drastic changes, and there are small sample and the small run to run variances. So small run to run variances can be handled with normalization, but the batch correction cannot be done but if you have got a huge amount of batch effect, then you cannot be done through the systemic and it should be done within the basis of the group subsets. So these are the two algorithms that are widely used in this kind of data sets, which are used for your batch correction. One is your lemma, which uses your two-way ANOVA to understand that what is your difference between the samples, the two batches, not the samples but the batches. And compact uses the empirical based method to make their medians equal and to reduce the technical biasness, or reduce those that has occurred due to the batch effect or due to the technical differences in your run. So this is how you will try to do a batch correction. But batch correction should be done only when you have no other option of trying to minimize that, and you have precious samples, or you want to have a very important experiment running. Otherwise it gives a lot of false signals that you need to finally validate before you can move ahead with it. So here, if you see in the left side in the box plot, so if you can see that one here, the experiment has been done accordingly is that after running all these samples, these two samples before running the run could have been completed before there was some issue with the instrument. So you can see clearly that all these samples are behaving differently. Their data distribution is different. So as I was mentioning, that all your data distribution, so that's what looks like the normal distribution, will not be the kind of that remains, that will vary if you keep on varying it across. So these two samples are your behaving differently. So now what can be done? So now batch correction will try to bring your median to the same. So after batch correction, so it looks like a kind of, here also you can see that your medians are same, but this is because to your normalization. So since normalization also tried to do it your so normalization also tries to remove your technical biasness, but it could not do much, and it was quite much evident because of your uh, because of your batch effect. So now after that you try to do it, then also you can think that there still differences remain. So this tries to keep your median in a same or the distribution in a same manner, but sometimes it becomes detrimental for the analysis, and it gives you a lot of technical uh, biasness. So but here another I think that you could not point out or could not understood. There's a the batch correction should also be done. For example, if you are running a pool sample, so if you can see that here these are the pool samples. So if you run a pool sample, so pool samples is your same sample. So why these experimental strategies are also required or is important is that, so if you have a pool samples and you are running each after each 5 samples, which is technically the same sample. So pool samples are made from all the samples that you are running. So it represents all your groups and all your samples. So if you are running those, so then that technically that same sample should behave in a same way. So this kind of normalization would have been done using, why I had given this example to let you understand that. So this how this batch correction should have been performed is that since the pool samples is the same sample and that has run across all the batches, and that has come fortunately that after since you are running a pool sample after uh, the end of the batch as well. So what is happening is that it is capturing all the changes that have been happened due to the technical biasness. So here what should be the thing is that the pool should have been normalized, should have been corrected, that this is my this is how my sample should look like, then this should have been brought accordingly. Otherwise what should have been done is that according to these pool samples, the rest should have been corrected. So this is possible with your combat. So in the combat what happens is that if you say that one data set if you say that my data set one is the one which is the correct, so data set one is behaving is correct, so the previous pool samples are correct, and you try to bring out my these two samples accordingly. So what will happen is that according to the pool samples, so it will bring similar to that sample. uh, similar to that pool sample. So then your batch correction is done correctly since you have the reference. So you are telling combat to this is my reference. Please do it according to this reference. Correct all my samples. So this would help you to remove in a much better way or much robust way. So this is what is so this is how we will so in the combat if you put on that if you run the combat, so it will automatically found out that how many batches are there on the basis of the variances and the covariances in your data set, and then it will adjust your data, and then it will fit your model, and then it will adjust the data and it will give you a corrected. So this is important step. So how it should look like is that once your data is this, so one batch and the two batch whose PCA will look like this, your one batch and the two batch, and after the batch effect removal, so it will all come in a simple way and it will all remain scattered, so which should be there. So this is how so combat helps in making that, which doesn't limit allow. So here again you will try to have an trade off that which algorithm to use for your according to your experimental requirements or your data's requirements and how it is done. So after so I think it's uh, clear, right? Or any question of there till the batch effect and normalization? No questions, right?
Can I ask you one question?
Yeah.
Can you move to the 22 slide?
Yeah. Yeah.
So, so both the column are after normalization, or what do you mean? Means both are written here after normalization. So I
Yeah, so
couldn't.
one is before batch corrected normalization and after is another is after batch corrected normalization.
Okay. And can you just elaborate what the pool sample refers here?
So pool samples refers here are those samples that have been taken from all the original samples that you are running, uh, an aliquot from all the samples, so that which you can run after each before and after each and every 5 samples, so that you will get a reference. So it is the technically a same sample which is being run to monitor your instrumental performance.
Okay. Means pool 1 containing all aliquot of my sample. Is this what you are referring
Yeah,
to?
so pool 1, pool 2, pool 3, pool 4 are same, actually same, and it
Okay.
contains from the samples from each of these means
Okay.
aliquot from each all of these samples.
Okay. Okay. Thank you.
Yeah. Okay, so now another thing that what I was what again I will refer to your fragmentation pattern and the according to your fragmentation pattern and what I said that so here we are what we get, so your fragmentation can be different during different time due to the different factors. As firstly I already mentioned that your fragmentation can be different then so here we get in the especially in our proteomics dataset. So if you get any omics dataset like the transcriptomics, but in the whatever mass spectrometry based datasets that we generate, be it proteomics, be it metabolomics, so one thing is that we get a lot of missing values. So this is largest number of missing values that are obtained are among these omics datasets. So this is because this is a stochastic process. What is a stochastic process? By the word stochastic process, I mean is that we try to predict here. So what we are trying to predict? We are trying to predict our ions, what we are flying in the mass spectrometer. So when the ions are getting detected or ions are getting uh, detected, so we are trying to predict what is the sequence of the peptide, what is the sequence of the proteins. So it might happen that some ions can get fragmented well in one one peptide can get fragmented well in one sample, in another sample. And also you have to make you have to understand that the process is quite it is not very much straightforward, where you have the process of your digestion. So in the one sample, your temperature when you are digesting in batches. So what will happen is that if one got a specific set of temperature requirements during digestion, another set of sample got another set of batch of sample got another set of uh, which did not give you the peptides, and you have got a lot of missed cleavages in your sample when the peptides the digestion was going on. Then there's a desalting. So in one desalting you have given some added extra pressure that leads you to the loss of certain peptides. So all these things can lead you to that there might be some fragmentation issues, and for which ions did not got detected. And also in the algorithm, in the passing the threshold criteria and all, it can lead you to the loss of those peptides or those ions. So for this, what will happen is that, so it will have a lot of missing values in your whole data sets, where our protein in the samples will not contain any values, or otherwise it will be missing. So now there is three concepts of the missing values. One is your missing completely and random. So now when you got the data set with all your proteins and you are getting the missing values, so what you will do? So now you have to take a decision, right? So if one decision can be that if my I have the peptides or I have the proteins, and I have the missing values. If for example, I have seven control, seven disease, and if any one uh, uh, place in any one sample that particular protein is missing, protein value is missing, so I will just discard it out. So I don't need that feature. This can be one approach or one way. But what will happen is that, since I told that the mass spectrometry sometimes it happens is that due to the fragmentation pattern or your there's uh, this your experimental pattern and all, it can lead you to the uh, might be there is some issue, and that's why you are not getting that protein's value, intensity value for that particular sample. So then you will be losing out the signals or losing out the proteins that can be of your interest or biological interest. So that's why we need to have the decision that when we need to when we need to take it out and when we can ignore. So another, was that thing what we generally, what is the practice is that, or what the generally it is followed by the proteomics community, or is that the generally if it contains more than 30% of the imputed missing values in any one of the groups or in the group sets, then it is generally discarded. But otherwise 30% data are generally imputed by the 30% are generally imputed by different algorithms and taken forward. So now another this concept is that missing, when we have the missing completely and random, then what is that, it is not related to any study variable, that whatever random, whatever proteins that we are getting, whatever protein value data is missing doesn't depend on any kind of variable. Rather, it is missing here and then. Sometimes it is missing, sometimes it is coming. So it is full at random, and we are not able to understand it. Then we can ignore this kind of missing values, that okay, it's coming full random and it has no relation with our data or our protein samples or our biological samples of interest. Now another thing is that the missing not at random. So this is what, this is systematically missing related to our unobserved data. That is, example is our low abundance peptides. So if our peptides are low abundant, then it can be it is coming or it is because of your, as I mentioned, your digestion and all those desalting procedures and all. Sometimes it is coming and sometimes it is not coming. So in some samples it is detected and quantified, some samples it is not. So these kind of peptides we should not ignore, or we cannot ignore completely. And these are but the in our proteomics datasets, we get this kind of unobserved data only mostly that is are missing not at random, and another is that the missing at random. So this is also one kind of thing which is due to the missing and the observed variables, that is like the peptide characteristics. So if you have, as I mentioned, that your side chains play a role in your peptide fragmentation and all. So if your peptides contain particular side chains amino acid in a mode number, then you are getting the values. So this is another kind of thing which is missing at random depending on the peptide characteristics. But most important that cannot be ignored is that the missing not at random when your low abundant peptides and all which are of your particular interest and which are not getting uh, which are not being quantified at in certain samples. So generally, as I told, that what it is done is that generally remove the variables or the features or your proteins with greater than 30% missing values across the variables. So in the sample set, if it is containing more than 30% of the missing values, so we generally what is generally followed is that the imputation should be done for only the 30% of the data, and the rest are filtered out. So this is how KNN missing value, and so the how, so there are different methods that you via which you can impute your data. One is you can use machine learning based different algorithms. So imputation is nothing. So what is imputation? Uh, if you ask, so if you have the missing value, then now how you will impute? So imputation, I think by the name you can understand that it is something getting the data, adding the data, that predicting the data from the other sets of the variables or other sets of samples, that okay, for this particular protein feature, these are the quantify that has been done in those samples, then what is my other, by in this my particular sample, what will be my value. So this is the imputation, the predicting on the basis of certain algorithms or certain features. So here, if we go, so this is KNN imputation, as I told that it is the widely used method to impute the missing values. So it can lead you to the imputation of the missing values for only a subset of variables for missing for each group. So the imputation, KNN imputation, should be used in a feature group wise manner. That is, for example, in a control data set and in the disease data set. So firstly, you have filtered out all those variables that are coming and that are showing your values or your variables that are much more having more than 30% of the missing values. So then what after that, what you will do is that you will take those control groups and do an imputation with those missing values or the features, so that it will get all the features nearest to it for that particular group, and then it will impute it. So here what you can see, that here green, this value is the missing one, and these are the values. So it will try to find out the nearest neighbor among each. So once it got it, then on the basis of this data, it will impute it. So how this distance or the who are the nearest neighbors are calculated? So mostly they are calculated with the Euclidean distance, that within this point data points, which one is the nearest one, and after you got the nearest one, the three so it will depend on how many neighbors you are giving. So k represents the number. So here if you say three nearest neighbors, it will take the data from three nearest neighbor. If you get two, then it will take from two nearest neighbor, and it depends on your data set and how you want to do that. So it will calculate on the basis of the distance, Euclidean distance, and then it will impute the value accordingly. So this is for the missing value imputation. So till now what we did is that we have still we are not going on to the details of that how the data distribution and how the look. So we have understood that once we have got the data and we have got the after all of these hurdles, when we have got the peptides and we have got the proteins, and now we are assuming it to be, and we are making transforming the data in a way that it is behaving in a normal distribution manner. And after we get the samples in a normal distribution and the normalized, so then but before that we need to have a see the check for the batch effect. If there are no batch effects, then we are going for the missing values. So we will do a imputation of the missing values, and then after log transformation and their normalization or the scaling, median mat scaling. So now we are ready to go for the statistical testing. So for the statistical testing, so we will, once we get time, we will discuss it in much detail. But here, for example, you know that mostly how we try to compare the means between your control and the disease state is that we, the basic thing is to compare the means of your variables of the control and the disease, that how different those are, and then to try to find out that how significantly difference your feature is. So here generally there is a the test that we use are the students t test. But what happens is that the sample size that we generally take off in the biological samples or the biological scenarios or the clinical specimens is that we are using a limited number of samples, and our so this leads to the decrease in the statistical power or the analysis. So we don't have sufficient, on the basis of the features or the number of features that we work on with, the number of samples that we take are generally very less. So maximum you can get 50-100 samples, but mostly generally in an ordinary average the number of samples that are being used are in the range of 10, 20, or 30 maximum samples that you can get. So here that happens is that the students t test sometimes it does not work well. So the moderated t test is another kind of t test which helps you to get the best result when it comes into that. So it is your similar to t-test, and it also compares the means of expression between the two groups. But difference, what difference, major difference lies or carries moderated t-test is that it lies in the calculation of your variance. So it so the variance, the way variance are calculated. So I hope that you all know the t-test, the formula that how uh, it is calculated. So if the calculation of the variance, it has that it students t-test calculate the variance from the data available for each protein, and it will moderated t-test will calculate variance from the selected feature. So this is only a difference between your t-test and your moderated t-test, which generally gets preferred when we perform with our samples in our. But here I would like to tell that there are various hypothetical testings and all those different t-tests and different parametric and non-parametric concepts are there. But generally what we uh, do is that we take the t-test and the moderated t-test to work. And next concept is that before I show some how to analyze the data, so as we are talking about normalization and those imputation and all, so I'll show that. So previous week we had seen that how the peptides could be identified from the ions from the chromatogram. Through mass spec we could identify the peptides. Now in this week we will see that how those data, uh, as we were talking about the concepts of normalization, missing value imputation, as we how we can do from uh, uh, using free software. So how we can perform those analysis that will help us in understanding. And also what I would recommend is that, so since we told just key and tried to give a definition or justification that this kind of normalization and this kind of things should be used in the proteomic dataset, but what you all can do is that you all can try out different methods and can see that the features or the proteins that you are getting on the basis of one normalization, so how much changes are you getting, so then only you can then you will get the answer that why certain normalization can only be used and not, and how we can go ahead. Now this is an important concept in our in any kind of omics studies, because here what we are trying to find out is that differentially expressed protein or a biomarker. So here if our protein or biomarker, if we are saying that it is a biomarker or it is a differentially regulated, but if it is not so, then what is happening is that it can be very much detrimental, because you are taking this protein or this RNA feature out, and you are this gene out, and you are trying to design an assay, or you are trying to design out drugs, or you are trying to knock out those and trying to find out the effect of those genes or the proteins in your disease state, but it is a false positive, then that would become very much detrimental. So that what is happening is that, so we need to have a FDR, or we need to control this false discovery rate. So this is what it depicts. So whenever we calculate the p-value, so it is not enough to it is the p-value is not enough to give our exactly to tell, or it is powerful enough to, and also on the basis of the number of samples that we use, that yes, this protein or the genes are your actually differentially significantly differentially regulated. So this FDR is calculation is required. So FDR threshold of 0.05 means that we are okay with our 5% false positive. So whatever proteins you are saying, or whatever genes you are saying, or it is significantly dysregulated, so then also you are saying that it might be that it has 5% false positives. So generally what is done is that after you calculate, so Benjamini Hodgberg correction is the one which helps in controlling the FDR values. So what was done is that individual p values are calculated for your using your t test or your moderated t test, and the lowest p value is ranked as one, and on the basis of the number of features. So for example, you have 5,000 features or you have 10,000 features, so you will divide it by the features, and then into the Q. So Q is your FDR threshold. It can be a 0.05 or 0.01, and then you will try to find out if it is exceeding or not exceeding, and then you will take. So this is mostly widely used in our proteomics datasets, or for the correction method, so that we can get the FDR adjusted or the adjusted p values, which is controlling the FDR in a somewhat manner. So it is more stringent than our p value, and we are less prone to get a false positives. So this is concept of the p-value and the adjusted p-value for the Benjamini Hodgkin correction, but there are other corrections that are again not recommended in the proteomics datasets due to their stringency and this. So we do not. So our major aim or the goal is to identify our proteomic biomarkers, proteomics markers, and for this we are trying to find out having the uh, after the sample processing steps and acquiring the data, so we need to extract out the features out of those particular data. So this is what we are getting. So now I'll just give quickly show the steps of the normalization, of transformation, and the missing value imputation, which I think you can uh, which you can think you can do is that you can uh, try to means I'll say that do it, just try to use it and try to find out certain things, that how it is changing your data. So here you can see your missing values. So if I just and here if you just do it, so you can try out different methods of finding it out, that how it is coming differently for your data sets. But here today we will show the different, so you can try out different normalization. But here what we will do is that we will just show till the normalization, and other things we will try out in the next week as we progress. So here this is the feature wise view of your before normalization and after normalization. So you can understand that how it uh, gets distributed. But if we look at our sample view, so here you can see, so this is your before normalization and your after normalization. So this data set is not so as I was mentioning, that every time you will not get your data set as it is. If in fact if you see that it is showing a kind of a normal distribution, so then you need to just go back, and you, if I do not perform any normalization, so all the time everything doesn't, it all depends on your data set and how you can reduce your biasness and the variances. Yeah, so if you see that your data set quite of the distribution becomes in the log transformation, it is giving your kind of a good Gaussian shape and distribution. So the reason for choosing this kind of data set is that you can understand in the real data sets it becomes quite difficult and quite this to understand that how to trade off between what factors to be used and how to make your data looks much more robust. So here you can see that the medians are not yet aligned. So now your question will come that what to do this with kind of data. For example, you have as we have mentioned that we, if we do a median normalization and scaling and all, this should get aligned, and the data distribution should look like a Gaussian distribution. So the here distribution, it is fine that sometimes the real data sets it doesn't happen. But if we look at here, then you can see that there is a quite a contrasting or marked differences, and when we are trying to normalize it, it is becoming very much distorted. The distribution shape is becoming very much distorted. Now the question comes that whether there are any problem. So you firstly, if you get this kind of data, so what are the steps that you should follow? The first step that you should follow is that if you get this kind of data, is that you need to go back and see that what was the run condition. The if you have, firstly you need to check out that how your sample was collected, how you have processed it, was there any issue on that time, was there any issue, for example this sample, so you can see, so if you are there any problem or was there any differences during the run time, did something happen that during the run was it did not occur, or the acquisition did not finish, or something happened, then is that so what are the differences? If you find out that there was observable differences during these samples, then what you can do is that you can just uh, move out, means just discard these samples and can then try out. And if it is not possible for you to identify that there was any difference or something, then you need to again look at the look at the, you need to revisit the sample type. So it can also be that what that, for example, if you are using a tissue sample or something like that, then it can be contaminated with blood and all those things that you have might have injected into it. and which has led to you to giving a different profile of your profile of the data, so that cannot be taken forward. So you need to go back and revisit various steps and various things. So this kind of data is quite possible in the real scenario, which you need to understand, and then you need to take a decision that whether you want to take it forward or not. Then you need to apply out the different normalization techniques. So or you need to remove out these samples if it contains a lot of outlier. And one concept that we will discuss in the upcoming weeks is about the correlation and all those. So if you find out that these samples are showing much different and behaving much differently, and its data distribution is much different, then what you need to is that discard these samples and then again perform the analysis. Otherwise, this keeping these samples and normalizing it and going ahead with the analysis in a very rough manner, it will, what will happen is that it will give a false result. So as we mentioned, so here also from here also distribution, you can understand that there is some issue has been happened with this particular sample. So as I was mentioning, that we are trying to here control our false positives. We are trying to find out the extract of the best features out of from the samples, keeping in mind that we are not going for any false positive, as well as we are not losing out any important feature or signal. So the I think now you all could relate and appreciate the fact that what we were studying as I started. uh, so that's why I start so there's we need to understand the your concepts of our fragmentation and what is happening really behind uh, before getting the data and analyzing the data. So now we can understand that there can be some multiple issues or multiple problems associated with it, which can lead to the generation of this kind of data, which we should go ahead and revisit and then only take a decision on whether we should take it forward or not. And also so in this software, you can try out different methods and can try out find out that with one method, with the set of methods that we were saying that it is recommended, like the median normalization and the knn imputation, what kind of proteins or what kind of markers you are getting as a differentially regulated. But if it is not so, and if you are using any other sets of normalization and any other sets of imputation, then what is the effect of that in your data set, and what type of data you are getting from that particular data set. So but all these things are in a real time real experimental scenario that are giving us all this kind of profiles, and all this we need to address before going for the further analysis. Yeah, so if anyone has any question regarding today's session or anything. Yes, any question?